In this video, we will continue our discussion about SARS-CoV-2 spike protein. Here, we will understand the study that has been done between intracellular chaperones, that is calnexin, and calreticulin, and the spike protein of SARS-CoV-2 or VLP particles, that is virus-like particles. So the study that has been done is to understand the role of calnexin and calreticulin in the formation of the spike protein, SARS-CoV-2 spike protein, we can say that. So let us start with the first figure. First figure shows about the this cell surface expression of spike protein. So this anti-flag tag has been used, and this panel A figure has been generated with the help of flow cytometer, and they have find out that this spike protein formation is almost comparable in case of this calnexin knockout or calreticulin knockout, and they are similar to the wild type cells. And when they did the same with the slight decrease in calnexin knockout, and when they did the same analysis for the other part of spike protein, that is S1 part, the RBD expression changes slightly compared to the wild type, and for calreticulin it is reduced more compared to the calnexin, as it can be seen in the live cells and also in the fixed permilive cells. And same thing is reflected on this ACE2FC binding, anti-ACE2FC binding experiment, that is the expression of spike protein on calnexin knockout is slightly less, and whereas for calreticulin knockout it is more reduced. So from this figure they can see that there is some role of this calnexin and calreticulin in the formation of this spike protein. So later on what they did is they did the co-immunoprecipitant study, in which they separate out this spike protein, and then check whether this spike protein binds with calnexin or calreticulin or not. So this first figure of the panel B shows about anti-calreticulin, and there are two parts. First one is the samples that are collected in the beads and the samples that are in the supernatant, and they can see that the spike proteins that are purified shows the signal for this calreticulin band. So this indicates that spike protein interacts with calreticulin, and same they can see in case of calnexin. The spike proteins interact with calnexin, and when they check for the presence of this S2 subunit, and they can see the S2 subunit both in the beads and also in the supernatant, though the amount of S2 in the supernatant in case of calreticulin knockout is very low. So later on, however, it is present. Later on, what they did is they check. See, from this figure, it is confirmed that calnexin and calreticulin interact with the spike protein. So later on, they did is they check whether the different mutants that they have produced by mutating the sites to change the glycan structure or to change the glycomics profile in the spike protein. And when they did the experiment for the calnexin, they can see that in each of the mutant, the calnexin signal is positive. This indicates that calnexin is interacting with This further substantiates their points that calnexin is interacting with the spike protein or S2 subunit S2 subunit. However, similar studies cannot be done for this calreticulin knockout because the molecular weight of calreticulin knockout and this S1 subunit is almost uh, comparable, that is around 50 kilodalton. A heavy chain of the molecular weight of calreticulin knockout and heavy chain of this RBD expression are almost same, that is 50 kilodalton. So from panel B and C, it seems more prominent that these intracellular chaperones are interacting with the spike protein and helps the spike protein to get their native structure. So later on they check the presence of this spike protein in different knockouts uh, through this Western blot. So in this they can see that in case of calreticulin knockout, the production of spike protein is less compared to the in this panel D, E, F. They see the how infectious the spike protein is from that are produced from different knockouts. And this they find out that the virus or VLP virus like particles produced from calreticulin knockout are low compared to the virus like particles that are produced in calnexin knockout, whereas in calnexin knockout it is almost comparable to the wild type. However, in calreticulin knockout, this S1 spike protein produced is very low. Later on, they did the viral infectivity test on of this spike protein, and they can see that for calnexin the infectivity is high compared to the calreticulin, and one of the reason could be that the virus that are produced in calreticulin knockout is low. And in order to check whether this is not the impact of CRISPR-Cas system that has been used to produce this calnexin and calreticulin knockout, they change the envelope of the spike protein to VSVG, and in this case they can see the increase in the infectivity of the virus. And because VSVG can has broad specificity and it can infect more cells compared to the this envelope that is present on spike protein. So later on they did the study on different kind of cells, or we can say they used different kind of virus, that is luciferase VLP system. So in this they can see that there is very less reduction in the S2 part of the spike protein production compared to the wild type. So one of the reason that is mentioned in the paper is that is because the VLP are produced from ERIC system, whereas spike proteins are VLPs are produced directly from ERIC. So whereas spike proteins are produced from cell membrane, or we can say they remove come into the extracellular matrix and have this cell membrane layers, whereas VLP whereas this luciferase VLP has directly originate from ERIC, or it does not have this cell membrane, whereas it has this ERIC uh, related membrane. So this could be the reason for this difference. Later on they express this luciferase VLP that they get from this system into the different cells. In this they see the reverse result, that is in the case of calnexin knockout they see the reduced viral infectivity or reduced luminescence signals compared to the calreticulin knockout, calreticulin knockout, they can see the infectivity has increased to a greater extent compared to the calnexin knockout. So this is the reason that they cite, is that because the luciferase VLP emerges from eric, whereas this spike protein emerges from cell membrane. That could be the reason for this. So that could be the reason for this contrasting results in case of spike protein and virus-like particles. So I hope you have understood this motivation behind this intracellular chaperones interaction and spike protein formation of the SARS-CoV-2 virus, and how they have carried out their study to resolve their question. If you need to understand this thing in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.